Hello, viewers. Welcome to BioDrunks. Let's learn some aspects of biotechnology.
How to draw an animal cell. So before knowing that, what is a cell? Cell is a structural and functional unit of life. And you have two types of cell. One is animal cell and one is plant cell. So in today's video, we are going to learn how to draw an animal cell. So first and foremost, draw some shape, circular shape like this. Okay. So this circular shape, outer boundary, is called as what? Plasma membrane. Okay. So first, we are going to draw all these structures, and then one by one, we will be labeling each. Okay. So whenever you're drawing, the most important thing is you need to start from the basic thing. So like we drew the circle here. Let's draw the most important part, which is the nucleus. So when you're drawing a nucleus, you cannot draw a circle there because this nucleus has nuclear pores and this nuclear envelope has pores in it. Okay, so always remember whenever you're drawing the nucleus, don't draw a circle. Here you can draw one dark circle, nucleus, and some structures like this. Okay, they are chromatids, chromosome DNA. Okay, now from this nucleus, you're gonna draw one side. You can draw like these structures, which represent endoplasmic reticulum. And always remember, these structures are going to touch your plasma membrane. Okay, try to draw it that way. So that is how, because endoplasmic reticulum, they are mostly related with transport. Okay, so here how you can draw this. So we are going to draw two ERs. Okay, one to represent a smooth endoplasmic reticulum and one to represent rough endoplasmic reticulum. Okay, now the structure is done. Now another most important thing is mitochondria. So when you're drawing mitochondria, draw a structure like this, and then inside you need to make cisterns like this. This is how the mitochondrial structure looks like. And don't draw one mitochondria, try to draw 3 or 4, because the cell has a lot of mitochondria in it. Okay, so try to draw 3 or 4 mitochondria. It would look better. So here how you can draw it. Easy. Now you have another important thing, that is Golgi apparatus. So that's how it looks like a cell phone. So in olden times we used to have such phones. So it looks like that. So just keep in mind, you can draw 5 structures, then make small vesicles like this. Done. Now we have a lysosome. So draw it in curved shape. So this is how it will be. A vacuole in a circular shape. So in animal cell, there are no more vacuoles. In plant cell, you have a bigger vacuole. So you can just draw. And you have ribosomes scattered on the cytoplasm and some attached to the ER. Now remember, the ribosomes that are attached to the ER, that ER is called as rough endoplasmic reticulum. And the ER in which the ribosomes are not attached is called smooth endoplasmic reticulum. So here how you can show the ribosomes getting attached to the ER and some floating. Okay, so these are the basic structures we have drawn. You can draw one more than one lysosomes. Okay, and ER vacuoles, lysosomes, mitochondria, Golgi apparatus. These are the most important structure. Now, labeling is very, very, very important. So let's learn how to label it one by one. So you have two types of labeling. One in which you can directly label it. Okay. Or you can do one more thing, that is you can just draw a line like this, label it as 2, and somewhere in the corner you can write 2 and you can write cytoplasm. Okay, so these are the two ways in which you can label it. we are going to go ahead with the first labeling. Okay, so let's see how we are going to label it. So the second structure which we are looking at is the cytoplasm. Label the next one as mitochondria. Let's label it. So here it is. Let me write it as mitochondria. See, always everyone gets confused with the spelling. MITOCHONDRION. This is a single one, so I'm labeling it as mitochondrion. If it's more than one, you can label it as mitochondria. Now let's label the next one. The next one, let's say it's vacuole. Okay. So let me write it. COULE. Okay. Let's write the next one. Let's label this. Okay. I think this is fine. Let's label it. So this will be your next, which is ribosomes. Ribosomes. Okay. Let's label the next part, that is lysosome. And then we are going to label the last parts. So, and always it's better to put it on the right hand side. It gives a very good view. Okay. And always on one side. Okay. So this is how it should be. So ER, let's write it here. This is the place where we are going to write ER. So this will be endoplasmic reticulum. Now, how would it be, smooth or rough? So as I have ribosomes attached to it, it will be your smooth endoplasmic reticulum. So here the ribosomes are not attached to it, so you are going to label it as smooth. And when the rear ribosomes are attached to it, it will be the rough endoplasmic reticulum. So this will become smooth. Okay, and let's label the last one. So this is gonna be your rough endoplasmic reticulum. Plasmic reticulum. Done. So this is how you can easily draw a cell.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.